Hello, students. Today, we will start with glycolysis. But before we move on to glycolysis, let us know what is plant respiration, since glycolysis is the first step of respiration. Plant respiration is defined as it is different from combustion because combustion is oxidative process, but there is immediate release of the entire energy, which is lost to the environment. Plant respiration, if you look at, is different from combustion since energy is conserved in the form of ATP. Now, where does this ATP come from? Or what is the substrate, initial substrate, which leads to ATP production? During the respiration, plants use carbohydrates. They may also use fats, proteins, organic acids for respiring and releasing energy or generating energy rather in the form of ATP. This leads us to the concept of RQ or the respiratory quotient, which is defined as the ratio between volume of carbon dioxide released by the volume of oxygen consumed. In case of anaerobic respiration, where there is no consumption of oxygen, the RQ would be 0. On the contrary, in case of organic acids, which have they are more oxygenated, therefore they require less oxygen to burn, and therefore the RQ would be always more than 1. So RQ is a significant estimation of the substrate which is being respired. In addition to ATP, if we look at the entire aerobic respiration, that is the respiration which occurs in presence of oxygen, we have the formation of water in the form of vapors. There is also production of carbon dioxide as a byproduct. The number of ATP molecules which are generated ranges from 36 to 38. This part we will talk about towards the end of electron transport chain, the last phase of aerobic respiration. When we talk about respiration, it can be broadly divided into 3 major steps. You have glycolysis, which occurs in the cytoplasm. Therefore, glycolysis occurs in both prokaryotes and eukaryotes. You have second major step in the form of a cycle, the Krebs cycle, which runs in the mitochondrial matrix. Finally, to generate ATP, we have the electron transport chain, which occurs in the inner mitochondrial membrane. Aerobic respiration, therefore, starts generally with sucrose or glucose. We would be talking more about glucose because finally sucrose is also burnt as a hexose sugar. It leads to the formation of pyruvic acid, a 3-carbon compound. So glycolysis is a 6-carbon compound. It gives us 2 molecules of 3-carbon compound pyruvate at the end of glycolysis. In addition, we also get carbon dioxide, NADH, which is the reduced cofactor, ATP, and water. This is followed by oxidative decarboxylation of the 3-carbon compound, pyruvic acid or pyruvate. This is an important step, an important intermediate compound, pyruvate, which cannot be easily broken down to acetyl coenzyme A, a 2-carbon compound which is fed into Krebs cycle. We require a multi-enzyme complex, also known as PDH. So, when pyruvate is converted into acetyl coenzyme A with the help of PDH complex, we also generate carbon dioxide and molecule of NADH. This acetyl coenzyme A is important molecule. Today, or in this topic, we would be referring to acetyl coenzyme A as a substrate to Krebs cycle for the release of ATP, NADH, FADH, and carbon dioxide when it is completely processed using electron transport chain. There are also products like FADH and NADH and ATP. But acetyl coenzyme A has its significance elsewhere as well. For example, it is also used to generate fatty acids. It is also used to generate certain hormones. Therefore, acetyl coenzyme A is not what we can say is the molecule which is fed into Krebs cycle, but it is fed into several other cycles or pathways, and it is known to be hub of activity. This visual gives you three major steps of aerobic respiration. As you can see, you have the first step, glycolysis, where you have breakdown of glucose to generate pyruvic acid or the pyruvate. In addition, these molecules are produced. You have then you have the Krebs cycle. Before the Krebs cycle and after glycolysis, you have the main conversion of pyruvate, the 3-carbon compound, into 2-carbon acetyl coenzyme A. And this is known as a link reaction because it links processes occurring in cytoplasm with those of mitochondria. Also, it is important step where a 3-carbon compound is pushed into Krebs cycle. So 3-carbon compound, how it is linked to 2-carbon compound acetyl coenzyme A. This step is also known as a Swanson reaction. So the mitochondria matrix shows two important steps. One is conversion of pyruvate to acetyl coenzyme A, and it also shows conversion of the running of the cycle to yield ATP, FADH2, NADH, and also generate carbon dioxide. It is then given into electron transport chain, which occurs in the inner mitochondrial membrane. So this is a gross picture. Today, children, we will talk about glycolysis, the first step in aerobic respiration and the only step in prokaryotes or anaerobic respiration. So let us move on to the next slide, where you can see the glycolysis along the steps, along with the enzymes and the intermediary products. If we see them closely, we will observe that glycolysis can be easily split into two major phases. There are total 10 reactions. At least 3 of them, these are important. So please note down that there are 3 steps which are irreversible. These are important. About them, we will talk a little later. And there are other steps which are reversible. So you have these steps, 10 of them. If we look at the middle of glycolysis, we can split this entire cycle in pathway, not a cycle, pathway into 2 phases. The first 1 to 5 steps, we can call them as preparatory phases. So these are preparatory steps which will consume ATP. Rather than giving ATP students, it will require ATP to run these 5 steps. So these 5 steps are they move from one intermediate to another, and you have the formation of glyceraldehyde 3-phosphate or dihydroxyacetone phosphate. Mind you, this is important to know that here, by this step, the 6-carbon compound has already broken down into 3-carbon compound. The next step, 6 to 10, is the forms the payoff phase. Why we call them as payoff phase? We call them as payoff because it is here that what you invested into in the first 5 steps, it is going to give you more energy. So it is cost effective. You get more energy. You get 4 molecules of ATP produced by the end of 10 steps. Since we have consumed 2 in the first phase of glycolysis, the net gain is only 2 molecules of ATP. We also should remember that water has been generated, and this is important here to note that pyruvate 2 molecules are formed because 6-carbon compound has broken down into 2-carbon molecules compound, and there are 2 molecules of 2 molecule of pyruvic acid, which is actually a 3-carbon compound. Glycolysis, something more about glycolysis I would like to tell you is that it is also known as EMP pathway, which stands for the 3 scientists who worked on it: Emerson, Mayrhof, Parnas pathway. So this is also the EMP pathway, glycolysis, as it is sometimes also known as. And we also have to remember that so many compounds which are formed here, most of them are phosphorylated. That is, they have phosphorus group added to them. Now, what is the importance of phosphorylated compounds is one important thought-provoking question which we need to answer. We also should know why glucose is so important, that sucrose is also broken down into glucose. From lipids also we generate glucose. Why is glucose important? So we need to work on why glucose is important and why the compounds should be phosphorylated. Let's just take up the phosphorylation condition. When the sugar or the moiety is phosphorylated, children, it is very easy for the phosphorylated sugar to be formed inside the cytoplasm, but it cannot cross plasma membrane, and therefore once it is formed, it is inside the cell, it cannot be lost. This is important. The second importance of this phosphorylated compound is that the activation energy of the enzymes which are involved in conversion, it is lowered down. So with less of energy, you can get more work done. The third important thing is that these phosphorylated compounds are known to generate ATP through a simple process where electron transport chain is not required. It is simply substrate level phosphorylation. So you get ATP formed while the electron transport chain can be omitted. So these are 3 points which you ought to know. Why glucose is used? The second question, we come to it. Glucose is an important molecule. As we know, glucose is used up in formation of storage molecules like glycogen, starch. It is also giving you pyruvate or pyruvic acid, which is the energy molecule when processed into aerobic respiration gives you a high number of ATP. We also know that glucose is a substrate or the product for many reactions, and it can be pushed into oxidative pentose phosphate pathway or OPPP, and you can generate again energy, and NADPH, which is important molecule for photosynthesis to happen. It is also able to generate certain extracellular matrix molecules like erythrose, suberin, and we all know, it forms the cell wall material by forming the glucose microfibrils, which give you the cell wall, which is so typical of the plant cells. Then, children, next would be our few more questions where we should know, once glycolysis has occurred, what is the fate of pyruvic acid? We should also know that if it is not pushed into aerobic cycle, how it is being processed in anaerobic respiration. The anaerobic respiration which occurs in absence of oxygen or in hypoxic conditions where the amount of oxygen is very, very low. We should also know that is there any difference between anaerobic respiration and fermentation, since fermentation is also anaerobic respiration. So what is the difference between two? We should also know that why is anaerobic respiration less efficient? It is only able to provide you 2 molecules of ATP, while aerobic respiration gives you 36 to 38 molecules of ATP. We need to also know how glycolysis is regulated. So let us look at these questions one by one. If you look at this visual, this slide, which gives you a gross picture of glycolysis, from glucose, you have generated 2 pyruvate. So pyruvate is there. The fate of pyruvate is given here. Fate of pyruvate can be either of three processes given here. One is this extreme end shows you simple aerobic respiration. We will talk about it later. We know that there is Krebs cycle involved. There is also the involvement of electron transport chain. If we look at anaerobic respiration or fermentative processes, pyruvate has 2 fates. It can be lactic acid fermentation, which leads to lactate formation. It can be ethanolic fermentation, which will give you ethanol formation. These are important steps. And if we look at these anaerobic processes, we can see that NADH is converted into NAD. That is, one is the reduced form, the other is the oxidized form. So pyruvate will give you lactate, and it will also lead to the oxidative form of NAD. Even ethanol will lead to that kind of a process. So let us look at this section of the slide where we have produced NADH through the fermentative or anaerobic respiration. But if we connect it back to the glycolysis, breakdown of glucose, where we followed the 10 steps, we should remember that we had produced NADH from NAD. So when these processes occur in the cytoplasm of the prokaryote, there is no supply of NAD. So to make NADH through glycolysis, the substrate is NAD. Where will it come from? It comes from these fermentative processes. So NAD, which is produced during conversion of pyruvate to lactate or ethanol, it is given back to the cytoplasm, and it is used up in the glycolysis pathway. So, in nutshell, it is a closed system. What is produced is taken back. It is not sent into any other pathway. So, it is therefore less efficient because it will simply become a closed system. This is important to understand. So the other thing which we ought to know is that is there any difference between aerobic respiration and fermentation? Yes, there is. When we talk of aerobic respiration, it does occur in some prokaryotes where you do not have the electron transport chain with oxygen as the acceptor, final acceptor, electron acceptor. You have some nitrates or sulfates. You have archaebacteria, you have purple sulfur bacteria. So they can produce, some amount of ATP without using electron transport chain. So that is anaerobic respiration. We should also know the difference between lactic acid fermentation and ethanolic fermentation. Mostly, the students are misguided by the fact that they have a concept that when ethanol is formed, it is fermentation. However, when lactic acid is formed, it is not. No, both are fermentative processes. The difference is that when we talk about lactic acid fermentation, for example, lactic acid formation rather, it happens in our body as well. You people have been told that when you over exercise certain days, you do some kind of physical activity or push-ups, your muscles show fatigue. It is because of the formation. You doing lot of exercise, breathing becomes less, oxygen available is less, so lactic acid is formed from glucose. Now lactic acid is slightly toxic. It changes the pH of the cytoplasm, and therefore the lactic acid accumulates and it leads to fatigue in the muscles. Whereas ethanol is better fermentation product because it is neither toxic nor it is going to stay there trapped somewhere. It is easily, it passes through, it is permeable through the plasma membrane. So ethanol is a better molecule to be formed. Moreover, our prokaryotes, microbes which are used in fermentators, are tuned to process the sugars to ethanol. So, we normally also say that ethanolic fermentation occurs in the microbes, not in our body. So, this is something we ought to know. Then we also have this. I've already told you why ethanolic fermentation is more efficient than lactic acid because it is non-toxic and can easily leach out through plasma membrane. We also need to know, children, that the beauty of the entire process of fermentation and glycolysis is this is very clear here. What I want you to realize is glucose is C6H12O6 molecule. So if you look at the ratio between carbon and hydrogen, it is 1:2. At the end of fermentation, what you get is lactate. What you get is ethanol. So there are 3 molecules of carbon and 6 molecules of hydrogen. Same is the case in lactate or lactic acid. So when we form these 3 carbon compounds from a 6 carbon compound, the ratio remains same. Ratio of C is to H remains same, and we produce 2 ATP molecules, direct production of ATP. Why I say direct production, I will talk about it a little later. If we look at this process now in little more detail, where we have the intermediates being formed, let us see what it is important. We have talked about pyruvate. Pyruvate is important, an important molecule because it is fed into Krebs cycle, and we all know that it leads to production of high number of ATP. Is there any other molecule which is important? Yes. Just before pyruvate is formed, we have the production of phosphoenol pyruvate, or more commonly known as PEP. This PEP is important that we have the PEP is important because PEP will give rise to, if we say pyruvate or pyruvic acid, either through using pyruvate kinase, where you have the ATP production. When you do not require ATP, then you can use PEP phosphatase and pyruvate is still formed. Pyruvate gives you acetyl coenzyme A, which is given into the Krebs cycle. Phosphoenol pyruvate is also a molecule which can lead to formation of oxaloacetate. We all know oxaloacetate or OA is an important molecule, a carbon compound, 4-carbon compound required to run Krebs cycle. It is the initial acceptor of acetyl coenzyme A, and you get the formation of citrate. Oxaloacetate can also lead to formation of malate. Malate is another molecule which is required for citric acid cycle. So this is how it is formed. Malate can be converted into oxaloacetic acetate or acetic acid in the mitochondria. Malate can also be converted into pyruvate in the cytoplasm, and pyruvate can be pushed into glycolysis. What I want to tell you is that all these steps are interconnected. Why? Because if you want Krebs to run, you should have an ample supply of OA, malate, fumarate. But if you take away, suppose you take away malate from Krebs cycle to form pyruvate, then this compound becomes less. So these pathways become therefore very important, where you have to have the formation of molecules, intermediary compounds, which when taken away from the cycle are supplied back also. This is a concept which is definitely important and should be understood as anaplerotic reactions, where the reactions occur to fill up reactions where they feed the substrate into the cycles when the substrate is taken away from the cycle for certain pathways. So this is what you must remember. Another important thing about glycolysis, when I said that it is direct formation of ATP, it does not require electron transport chain. Therefore, glycolysis becomes a conservative pathway since ages, since the day life originated and bacteria were formed. They all have been running glycolysis unchanged. And you have the formation of phosphoenol pyruvate. I give you the example of the last step where ATP is formed. There is pyruvate kinase, an enzyme, and you have phosphoenol pyruvate, a 3-carbon compound attached on one side of this molecule. The other site is occupied by ADP. Now, ADP is here and in close proximity of phosphoenol pyruvate. So during the vibration or the movement of the enzyme, there is a high energy phosphate in phosphoenol pyruvate. I told you that phosphorylated compounds are important. So this compound gives its phosphate group to ADP, and ADP now has one more phosphorus group, and therefore it becomes ATP. This is substrate level phosphorylation, very different from phosphorylation, which occurs through electron transport chain. Two major differences are you do not need any mitochondrial or any organelle or plastid to run the electron transport chain. Therefore, it can run irrespective of the fact the plant cell is a prokaryote or a eukaryote. Another difference is that you do not require oxygen as the final acceptor. You do not require ATP to run the show. So this is what is important. This is now the gist of the aerobic respiration. We have already done the first step, first phase of respiration, where we have broken down glucose into pyruvate. Lactate we will ignore here because we are going to push pyruvate into mitochondrial matrix. In the next few lectures, we will talk about Krebs cycle. The Krebs cycle runs in mitochondria. Mitochondria, if you look at, has matrix. It has these foldings, and we call them as cristae. And you have pyruvate being converted into acetyl coenzyme A inside the matrix. And then it is pushed into Krebs cycle, which also runs in the matrix. Now, what is important here, I should tell you, most of the enzymes are present in the matrix. I want to highlight one important thing, that what will happen if the cell has lot of demand, or the plant has lot of demand for ATP, and glycolysis occurring in the cytoplasm is not enough to take care of or meet the demand of the plant for ATP. Children, there are additional enzymes present on the surface of the mitochondria, and they also indulge into glycolysis of slightly different type, but it is basically the same type, substrate channeling, where you need the substrate only to produce ATP. So if you go with the concept that glycolysis occurs only in cytoplasm, it is a little wrong, because it also occurs on the surface of mitochondria, and few steps of glycolysis, if you can recall, occur in the plastid also. So this is one thing you must remember. Then we should also know that it is an important process, glycolysis, where we need to know that what will happen if too much energy is produced at the end of aerobic respiration, that is electron transport chain. What will happen? Plant does not need, and it is producing energy. Energy, if produced in large amount, may be destructive to the system itself. It has to be stopped. So if we need to stop, okay, you will say do not run electron transport chain. But we do need to have NADH produced because it is required. We need to go back and have Krebs cycle running because we need to push citric acid to the formation of fats. We also need to form pyrimidine and purines. We also need to form sugars. So we cannot afford to just stop the cycle. Moreover, stopping machinery will also lead to some kind of complexities in the metabolic pathways. What we have to do is we need to regulate that. Partially, we can run the cycle, we do not produce the energy. So, keeping this concept in mind, we move ahead to regulation of glycolysis. Now, children, regulation of glycolysis occurs at majorly two steps, and there is a third one also. The primary regulation is do not let pyruvate form. So I will not let pyruvate kinase, the enzyme which converts PEP into pyruvate. So let me stop pyruvate kinase not to catalyze the process. I need to check pyruvate kinase. That is why I already told you the importance of PEP. PEP is required, we can push it elsewhere, but pyruvate kinase has to be stopped. What stops pyruvate kinase? Simply lot of ATP molecules available in the cell will tell pyruvate kinase, you need not catabolize now pyruvate because we do not want ATP to be produced. As simple as that. There are other molecules also which are involved. So this is major step where you can check glycolysis. The other step is here where you can check the phosphofructokinase, the enzyme here, phosphofructose kinase, which converts glucose into glucose 6 phosphate. And let me tell you, this is the second important enzyme which we can regulate. So we will not allow the formation of a particular intermediate, and phosphofructokinase is allowed to be inhibited if need be. It may be activated if the need be. So this is another step, and all these steps are irreversible. I told you there are 3 steps at least out of 10 which are irreversible. The third is the first step, hexokinase. And remember, in these steps, only ATP is produced. So hexokinase. So hexokinase is another molecule which can be inhibited or activated depending upon the demand of the cell. Therefore, if we look at the regulation, we have to go back from the 9th step, we move to the first. This is known as bottom-up regulation because you go from the bottom of the pathway to upwards. So it is bottom-up regulation, and if you have large amount of ATP being produced by the cell, it is taken care of by the system of the enzymes. Let us see what inhibits them. Very clear, 3 sites of regulation at hexokinase, phosphofructokinase, pyruvate kinase. If we go bottom up, pyruvate kinase is the primary step of regulation. Then comes the PFK, and finally hexokinase. These are inhibited allosterically by its substrate, pyruvate, and the production of ATP. If ATP is too high or the amount of pyruvate is too high, pyruvate kinase will be automatically inhibited and a PEP will be accumulated. But PEP, as I told you, is a molecule which can be taken elsewhere. So it is clear that there is no accumulation and no toxicity spread. Krebs intermediates also inhibit pyruvate kinase. Secondary control is that PFK and a PI will activate it, and the substrate, if you have the product PEP formed, it will inhibit. One more control is at hexokinase, and it is given as in some cases, it can be shown to inhibit the process, so, or regulate the process. So this was all about glycolysis, and we will surely move on to the next one soon.